I have the pleasure of introducing our panelists from the research working group. Of course, it's going to be Dr. Greg Allen and Dr. Vickie Blazer. And for those of you who are not as familiar with Dr. Blazer's work, Dr. Blazer is a research scientist with the U.S. Geological Survey's National Fish Health Research and Laboratory at Leetown Science Center in Kearneysville, Virginia, West Virginia. With a career that includes teaching and conducting research in fish tissue, fish pathology, and fish nutrition, she continues to work with graduate students at West Virginia University, Hood College, and Penn State, and teaching classes through the Fish and Wildlife Services National Conservation Training Center. Her current research interests involve understanding fish health and its relationship to ecosystem health, environmental contaminants, pathogens, parasites, and land use, and the sources, pathways, and effects of endocrine disruptors and other contaminants of emerging concern. Dr. Blazer received her BS in marine biology and a PhD from Rhode Island University and completed a postdoc at the Veterinary College at the University of Georgia. So I'll turn it over to Dr. Blazer and Dr. Allen now.
Okay, thanks, Bonnie. And I'm going to get us started here by just saying a few more words about the report that we are finishing up. Our research and data gaps work group decided that one of our roles is to capture current and valid research findings and communicate them in various ways, and including to our other work groups that we have in the project, so that we're all staying up on some of the latest information. So we're going to give you some key findings, identified data gaps, and a few thoughts on future direction.
Okay, I'm going to take over for the key findings. As Greg said, this report, we did focus on literature that focused on the Chesapeake Bay watershed, but obviously we looked through literature, globally as well, concerning pesticides. One of the key findings here in the Chesapeake has been that while pesticide use in recent years has decreased in mass, or the pounds used or applied, toxicity has increased. And this is particularly true for some of the lower trophic level groups, such as insects and crustaceans. The other thing that's probably true all over is that pesticides are always found in mixtures. And the graph here just shows 3 different herbicides, and this was actually monitoring done at Antietam Creek. And what you see is that often all 3 of those are peaking at high levels when one of them peaks. It's often combinations. Next slide, please. So multiple studies point to the importance of mixtures and the effects, the ecosystem's effects of those mixtures. Some of the important things are that when those lower trophic level organisms are harmed, ecosystem impacts can carry all the way up the chain. Degradation and nutrient cycling mechanisms can be disrupted by numerous pesticides. And then, as we've heard about PFAS, some of the non-lethal effects, such as immunomodulation and endocrine disruption, are observed in animals exposed to pesticides. Next slide. The other thing, as we've heard about in terms of PFAS, is pesticides also can be uptaken and travel through organisms to various tissues. And they often end up in, for instance, the flesh that's used for food, but also parts of organisms that might be used for medicines, and serums or plasma that might be used both in medicine and in research. Some of the good news is that there is evidence that some best management practices do work for reducing exposure to pesticides and estrogenic compounds. Next slide. And then we've all heard a lot about PFAS, and this sort of gets us into key findings as well as data gaps. We certainly have evidence here in the Chesapeake that we are finding PFAS in surface water, in fish and other organisms. So we know they're there, but there are a lot of questions yet as to where they're coming from, and also certainly effects. Next slide, please. So as far as data gaps, some of the things we need better documentation and monitoring to really understand temporal changes in pesticide occurrence and exposure. This is the same slide I previously showed. And I put it up because it really illustrates the difference both monthly and annually when we monitor for some of these things. So for instance, in 2014 and 2016, you can see some really high peaks, a little less high in 2015. And then in 2017 and 18 lower, and then a little higher in 2019. So a lot of variation. And we need to understand how this is influenced by things like best management practices, land use practices, as well as climatic factors that might be regulating the occurrence and exposure. The other thing that's key in this is understanding exposures during key developmental times and sensitive periods in an organism's life. So for instance, many of these peaks are in the spring when eggs of fish and amphibians might be present, or when young of the year that are going through early development. And we know that those are very key sensitive times. So it's important. We need more information on those exposures. Next slide. And then the effects of exposures to complex mixtures. We've heard about all the PFAS, that the different compounds that are present, and it's the same with pesticides. There's lots of mixtures. And then it's not only the parent compound, but also the degradates. And we have very little information about what the effects are of some of these degradates and how they interact with other chemical contaminants. And then as, again, effects of ingredients other than the parent compounds that might be present. So again, the graph here is information from Antietam Creek. The first one on the top, what we're seeing is that most of the time we're seeing much higher concentrations of one of the degradates of atrazine than the parent compound. And then the lower is, the concentrations are less, but again, certainly some are high and they vary over time. So we need really to understand, are there synergistic effects, antagonistic effects, additive effects? You know, how are these affecting organisms from lower trophic levels up to fish and ultimately humans? Next slide. And then the other concern is species sensitivity, and that leads into ecosystem level effects. Most laboratory exposures are conducted with model species that may or may not have similar sensitivities as both economically important populations and also as some of the key food web species. And that basically, when you look in the literature, you tend to see a lot of contradictory studies. And one of the reasons is that using different species, you get different results. Using different life stages, you get different results. And so we really need an understanding of that, and then a better understanding of the food web effects and how things at the lower end of the food web affect those of us at the higher end of the food web. Next slide. And I think Greg is going to cover this part.
Yes, thank you, Vickie. So Vickie just described some of the top messages in this draft report. We were using a 10-year time frame. Not sure if I mentioned that before, but we were looking for peer-reviewed articles over the last 10 years to derive some of these recent findings and some data gaps. But here we're providing some thoughts about future direction for the research and data gaps work group. And 3 here just to mention. First, staying current on methods that are developed to assess risk from multiple contaminants. And this is for pesticides and their degradates. It could also, of course, be used for other contaminants should we get to a place where we have a method. And so we plan to discuss and contemplate a next report that would be something of a roundup on what do we know about methods for this concern. And I've got a link here for a study that was something of a meta study looking across all sources of journal article and other literature. And it's really an impressive summary of where things stand. It's an example of one source of information that we might use to, again, give us a snapshot of where we stand with this. This is a very long-running issue and challenge, but there is work that's being done. I've recently seen some grant programs that are giving out money for projects that are designed to help move us forward with this challenge. And this is from Organization for Economic and Community Development, I believe, in partnership with the United Nations. Yeah, so it's a really worldwide synthesis on where things stand with methods for assessing risk from multiple contaminants. And we know that in our surface waters in Chesapeake Bay, there is always some combination of things present. And our standards, our risk criteria that we have for some of our contaminants of concern are all based on that individual contaminant. But when we think that there might be synergistic effects from a cocktail, as some researchers refer to it, a cocktail of contaminants, then the question is, are those single contaminant risk criteria still protective in light of there maybe being some synergistic effects? So those are some of the challenges around this that have been ongoing for a very long time. And we may decide our next report will be some sort of synthesis on where the state of the science is. Next, as we have already talked about, and we, I guess, made a promise to Dr. Peasley, we want to keep up on all the latest science with regard to PFAS and pesticides. That's important for the environment, for the living resources of the Bay. It's also important for our farmers and for healthy farms and healthy soils. So this is something that we will certainly be keeping up on, and we'll see how our work group can help to pull together summaries on this topic and keep us all informed. And then last here, I mentioned that last year we did a large-scale Jamboard, and we're working through the feedback that we got. And it's very possible that our work group will have some suggestions coming out of all of that input that might give us good ideas for future work. And get the emphasis here on trends. I think our research work group can also help keep up on what do we know about trends, and that's trends in concentrations of pesticides in surface waters, in fish, and other living resources. Trending is its own science that can be challenging because you have to have enough data to really be able to make a statement about a trend. But it's so important to us all to know which direction we're headed, that I think that's another place that our work group will be thinking about adding value to this community. So those are a few future directions for us. And that's most of the comments that we had for this report and some of the future work for this particular work group.
Wonderful. Thank you so much, Dr. Blazer and Greg. And we are really excited. The work group has been putting a lot of great materials together to look at for research. We've actually added a bunch of studies, which is partly why we've delayed publication. So we'll be looking forward to getting that out the door. We have some questions for you guys. One is, what sort of BMPs were documented to improve contaminant levels in water?
Yeah, well, let me start and then I'll give it over to Vickie. Yeah, so among the universe of best management practices, and particularly those that we track in the Chesapeake Bay Program Partnership, mostly for the purpose of nitrogen, phosphorus, and sediment reduction. But among that universe of BMPs, we don't have a whole lot of data about effectiveness for the contaminants of concern, most of which are organic chemicals, meaning they have carbon in them somewhere. We don't have nearly the amount of data that we'd like to have, but we make some assumptions. And one that comes to mind is that wherever a BMP is highly effective at trapping sediment, it probably gives us a benefit on a range of organic contaminants because they tend to be sticky to sediment, carbon to carbon sort of attraction there. And so wherever we capture sediment, we're probably doing something good for the loading of toxic contaminants, and also for the contaminants that are in rainwater that get dissolved or get mobilized in stormwater. Wherever we have a BMP that allows that stormwater to infiltrate, to settle, to have time to be biologically treated in the soil zone, things like green stormwater practices, green ponds, and rainwater capture devices that allow that surface water to percolate, that is also helpful. So we end up having to apply some of those broad assumptions for the most part because we don't have a lot of data. Now, one that I will mention that we have some data on that's not pesticide specific, but I think we had this presentation at this conference a few years ago, manure injection. So there's a BMP that instead of applying manure broadly to the field, if we use the injector technology and get it down into the soil zone, we see that there's a lot less runoff of things like hormones that are in that manure. So that would be an example where we have some specific data.
Great, thank you.
And so, Vickie, do you have anything to add to that?
No, I agree. I think a lot of the BMPs like riparian zones and things like that cut down on runoff from fields into streams and rivers help a lot. also, I guess we should mention some things like cover crops. If you're then killing those cover crops with herbicides, that's sort of a negative. So that's why we say some BMPs. So just something to think about.
Good. Okay. We have another question. Why are degradates important?
I'll take that one if you want, Greg.
Yes, Vickie.
At least from the aspect of fish health, there are some papers out there that have shown that some of those degradates are just as toxic, and in some cases more toxic than the parent compound. And so one of the issues is that we're often not measuring the degradates. We're only measuring the parent compound. And in that study we did from 2013 through 2019, we only started measuring the degradates in 2018. And you know, the data that I showed is we're seeing often higher levels of the degradates than we are of the parent compound. So what we really need to know is which of those degradates have adverse effects on various organisms.
Okay, thank you. We have another question. What is the practical application if we have a method that would assess risk for multiple contaminants?
Yeah, I think that's a great one and a challenging one, but I guess if we were to get to the point where we had a valid method, it could be used in predictive tools. So it's conceivable that we could use information like land use and monitoring data, actual monitoring data, maybe other information, some combination of those things to say, given what we now know about risk from mixtures, we are likely to see in a given area a higher risk to living resources than in others. So ideally, we could roll it into some form of a predictive tool that would then allow managers to go apply management practices in those areas to reduce risk. So that's a little bit of a visionary outcome. But I think the question of if we did get to the point where we could put a number of detected contaminants, maybe even some of those degradates that Vickie just said maintain some toxicity, into some sort of risk analysis tool that we can then use it in a way that actually helps us with management actions and decisions. And we'll just have to see. Right now, we have some of those kinds of predictive tools, but they are not really considering synergies and lots of contaminants being present. So we'll see if we can get to that point.
Okay, thank you. Just wondering if there is, any pesticide monitoring that's going on in the bay waters that you all are aware of, and if so, is it enough?
Well, I'll throw a few things that I'm aware of and see maybe Vickie has something to add. With regard to toxic contaminants in the Chesapeake Bay watershed, we don't have nearly the amount of data that we'd like. A lot of the resource goes to monitoring for impacts of nutrients and sediments. One of the very few long-term data sets that I'm aware of for pesticides comes through USGS and their river input monitoring stations, RIM stations. And so these are located in some of the highest flow rivers to Chesapeake Bay. And for quite a long time now, there's been a standard set of pesticides that have been analyzed at those rim stations. That's a good example of that future direction that we just mentioned of looking at trends. I would guess that that's one of our highest potential data sets to use for trends. We really should get in touch with that data and see if it's useful in that way. Other than that, I don't know of much long-term monitoring. It tends to happen, excuse me, more in a snapshot sort of basis, in federal studies, maybe some state monitoring, and maybe some academic work. And in those areas, we do get some snapshot monitoring, but they tend to not be long-term. Does that match up with your understanding, Vickie?
Yeah, I would agree.
Okay, great. Thank you. Well, it sounds like we have a lot of important work to do ahead, and this report will really help define some of those data gap areas, and it's going to be very exciting to see that. It looks like we have 2 more minutes, and there's a question here. Has correlation of land application of sewage treatment plant biosolids and pesticide contamination levels been studied?
Yeah, not to my knowledge, but that doesn't mean that there isn't some data and some papers out there. I will say that biosolids seems to be coming back on the radar brightly again, because we have some recent data that shows that PCBs and PFAS are partitioning over to solids in our wastewater treatment plants. And in a lot of cases, we end up with land application of those biosolids. And that is some just very recent data showing that there is a fairly strong signal and amount of PCBs and PFAS in those biosolids. Whether it also includes pesticides, I'm not aware of data, but not really certain of the answer there.
Can I add to that, Greg?
Sure.
No, you're absolutely right. There's a strong correlation between biosolids and PFAS. I am not aware of anybody doing insecticides at the same time, but the PFAS definitely correlate with biosolids. And in many states, Maine and Alabama and a whole bunch of ones, there have been big studies on that. It's good.
Yeah, and some tragic outcomes. I think you mentioned dairy cattle that have had to be slaughtered as a result of being so contaminated that they were not able to provide any product anymore. So we know that biosolids that are land applied on farm fields can have some really dramatic impact.